Thank you so much for the invitation to speak. It's a real honour to be here today, and what a wonderful conference. It's my first time. So the human brain is staggeringly complex. We are all united here today, even if you don't work with mice, by the greatest challenge, which is to understand the human brain, both in healthy and also in unhealthy clinical populations. But I'm here to tell you about mice. Let me start by turning back the dial over 100 years, and I'll tell you a story of Abby Lathrop, the mouse woman from Granby, and you can see her house here. Abby became a rodent breeder after a failed attempt at poultry farming, and I'm not sure we need to know any more about this. But initially, Abby bred fancy mice, and an example of one of these is the Japanese waltzing mouse, and this mouse spins around, and collectors would purchase these mice from her. This mouse in particular was quite popular because of its spinning, but also because it was very rare. And it was rare because it was largely difficult to breed. Abby ran a bustling business, and at one point she housed up to 11,000 mice at her farm. She was clearly an entrepreneur. So why am I telling you about Abby? Well, Abby took a very detailed approach to her business, and part of what she did was take detailed notes, and that's in order to be able to get these really difficult to breed mice to actually breed, and so she could make more money. But she was also a budding scientist, and she reached out to a pathologist, Leo Leob, at Washington University. And as a result of her notes and her interest and her energy, the two of them together created mouse models of estrogen-fueled cancers, and they published 10 papers together. So despite a lack of university accolade or even connection with the scientific community, Abby became a mouse modeller, which is the theme of my talk today. A connection, she gave a mouse number 57 to the two geneticists pictured up here, William Castle and Clarence Little. And this mouse is now the mouse that I have spent 20 years working with, and it's the C57 Black 6 mouse. Some other fun facts about this mouse. They were the second animal to have its genome entirely sequenced, and also 10 years ago, they were flown into space by the Russians. So mice have been integral in helping us to understand human disease mechanisms in the past, and today I plan to show you the utility in understanding complex cognition. So as I mentioned, I'm a neuroscientist, and I've worked with mice for over 20 years. The animal model field frequently gets a bad rap, and I'm not going to avoid just criticism here. I've conducted almost every behavioural test under the sun, and honestly, most of them are not optimal. Some of them are 5 minutes long, and mice are fickle creatures. They're sensitive to a bump in a neighbouring test room, or a mobile phone, not on silent, or even a sneeze. Many of these tests evoke stress, or they do not measure the cognitive construct that is relevant to humans. So take the water maze, for example, and please bear with me here. The water maze is a very characteristic test of learning and memory. It was devised 30 years ago by Richard Morris. The mouse is placed in a circular pool, it's filled with water, and their swim path is tracked. The mouse must use external cues external to the pool to navigate to a submerged platform, and typically escape times will decrease with training. This test is sensitive to hippocampal dysfunction, and it is widely used to identify cognitive decline in Alzheimer's mouse models. So during early learning, the mouse will circle the perimeter of the pool, and if it finds the platform during the trial duration, it will be a fluke. But once they've habituated to the water and realise that there is an escape, they change their swim path to serially search the pool, sweeping back and forward until they bump into the platform. It's during later training that mice shift their strategy to use the cues external to the pool, and they eventually swim directly to the platform. If a new drug improves memory in an AD mouse model, Alzheimer's mouse model, will it work in the clinic? Is the drug really working how we want it to? Are its effects on lowering anxiety or boosting swim stamina? Patients are diagnosed by clinical assessment and tasks that test verbal recall. It's pretty clear that there's a large discrepancy between these models of assessment. So it's not that we can expect a one-to-one, because obviously mice and humans are different, But we can aim to develop tests that engage similar neural substrates in both humans and mice, and ultimately this would enable us to better predict efficacy across species and therefore improve the chances that a treatment that works in mice will also work in humans. So one approach is to put patients in a maze. Here is a human version of the water maze. Many water maze analogues in humans have been trialled, but protocols vary and do not always correlate with the species. Poisson and colleagues were the first to identify how well a human version of the water maze compared to the rodent version. They moved away from traditional measures of learning, applied the best of both worlds of research, and came up with a template for comparing the two. Their work is a lovely demonstration of how translational behaviour can be done. However, some key differences still remain. And these include, motivation, also most certainly stress, physical activity required to complete the task, and also the fact that mice were still woken up in the middle of their sleep cycle to swim in a large pool. So what does it look like if we put the mouse in the clinic? So I train mice to play computer games. These games are based on computerised tasks used for assessing cognition in humans. Mice are rewarded with strawberry milkshake for correct nose pokes to the screen. And this mouse here is excelling at the rodent version of the CANTAB paired associate learning task, which is a task sensitive to early Alzheimer's disease. CANTAB is a computerised assessment battery widely used in academic, clinical and pharmacological research. So the touchscreen method was pioneered by Tim Bussey and Lisa Succeeda, who are psychology trained and saw a solution to the translatability gap between animals and humans. There are now multiple CANTAB-inspired mouse tasks and over 300 sites using these chambers around the world, shaking up the way that cognition is modelled in mice. So let me take you through a touchscreen setup. Touchscreens allow display of near identical stimuli and timing to human tasks. The screen can be partitioned into smaller windows to display stimuli and capture nose poke responses. In addition to the touchscreen, infrared beams provide a constant stream of data on the mouse's location and speed. Our data consists of location, timing and nose pokes, and we use statistical models to capture responses over time and over multiple trials and sessions. If you happen to possess many touchscreens, high throughput testing is possible. And this allows multiple animals to be tested at the same time. So this is important, as I mentioned, mice are fickle and sensitive to environmental changes. And we know that cognition varies with the circadian rhythm. As each mouse requires 1 hour of testing per day, it's useful to test mice close in timing to each other. Also, our students are very grateful to leave the testing facility before dark. So this is a game changer for reproducibility and for mental health. Importantly, each animal shows up to their task willing and without stress. An unwilling mouse can choose to sleep or simply investigate the chamber, and accordingly, we choose not to capture their data. This is a very rare event. Mice are very motivated by milkshake. So how do we train mice to learn these games? The classic model of cognition describes an input that is typically sensory in nature. And this input will undergo computation in the mouse's brain and will rely upon these underlying cognitive concepts that my team and many others are trying to tap into. We use motor output, or a nose poke to the screen, as an indicator of performance. And we reward responses with strawberry milkshake and punish incorrect responses with light, and mice do not like this. This outcome is critical to allow the animal to change or to hone its behaviour. And ultimately, this motor output data is transformed to the measures of the cognitive construct that we're interested in, and I'll talk about this a little in detail later. So mice are not humans, and this task is not an exact replica of how cognitive assessment is run in the clinic. When humans are the subject, the reward is rarely a milkshake, and the motor output rarely involves whole body movement like it does in mice. Mouse vision, of course, is very different to ours, and we ensure that mice can discriminate between stimuli before we use them, i.e. they can see it. So how your subjects show up, if they do, and their demographics will impact the outcome of your study. We can control when our mice show up, their prior experience, their genetic background, and also who they live with. But a major difference is that we cannot verbally instruct mice on how to complete the task, and thus they require extensive training. Many touchscreen tasks have been created to replicate traditional behavioural tests, which you can see here. Many of them also have a human test equivalent, but not all do. My team have applied touchscreen tests to characterise complex cognition in multiple disease mouse models. However, today I will focus on one model because it allows me to take you on a journey to show you what is possible in mice and also tell you a few cautionary tales. So animal researchers and those working across species are here today at OHBM. And comparative neuroimaging is a powerful approach to resolve human brain structure and function. While it's important to invest resources into improving tools to resolve the structure of the human brain, if animals are also used to do this, we must ensure that our mouse models are robust. It's also important to consider if we're measuring the same construct across species, but how do we test this? And many of you might be wondering, are mice really smart enough to take on some of these cognitive tasks? I will now tell you the story of a mouse model of autism that carries the R451C mutated neuroligin 3 gene. This mutation was first identified in 2 autistic brothers. Introducing a rare mutation like this one to a mouse and then observing brain and behaviour is a common approach for understanding how that mutation may impact the human brain. And this is the power of animal model systems. The neuroligin 3 R451C mutation is an arginine to cysteine substitution, and it resides in the exon 7 of the gene. NL3 is normally trafficked via secretory pathways and inserted into the postsynaptic membrane. Here it binds with presynaptic its partner, neurexin, and it plays a role in maintaining the postsynaptic integrity. It also recruits glutamatergic receptors, which impacts on postsynaptic signalling, and it also has a role in signalling through the presynaptic neuron via its binding partner neurexin. When mutated, however, 90% of NL3 remains in the ER and is eventually degraded. The remaining 10% is inserted into the membrane. However, altered binding to pre- and postsynaptic partners has been identified, particularly with neurexin, due to the mutation's location in exon 7, which codes for the binding site between these 2 proteins. So this is an overwhelming diagram. The point is to show you that the R451C mutation in neuroligin 3 is not alone. Many mutations have been found to disrupt synaptic signalling in autism and are hypothesised to affect a common pathway. So introducing the R451C mutation into a mouse represents a valid way of investigating this common mechanism. Importantly, neuroligin 3 is a high confidence candidate compared to the others. There is a homologue in the mouse, and in fact there's 98% sequence similarity between mouse and human. So in order to model autism in a mouse, one must understand the phenotype in humans. And many of you will know autism is a complex neurodevelopmental condition with a range of features and related conditions that vary in phenotypic strength across individuals. Despite their name, secondary autism characteristics can be just as impactful to the individual as the core characteristics used to confirm diagnosis. And today I will tell you a story of how we've been focusing in on attention. So early characterisation of the NL3 mouse focused on social behaviour and differences in excitatory and inhibitory signalling in several brain regions. Presence of restrictive and repetitive behaviour have also been described. Animal models typically embody these core diagnostic criteria of the condition, and this is termed face validity. Important to note, actually, that not all studies have replicated the social cognition differences in this mouse. These are the tests that are simplistic and they suffer from lack of reproducibility that I mentioned before. They also do not capture human relevant constructs as they are based on ethological relevant behaviour. But perhaps this might be the best we can do concerning social behaviour in a mouse. Atypical cognition most certainly features in autism and interacts with core traits. Reports of atypical attention in infants who are later diagnosed with autism suggests that this difference could potentially be driving the development of core traits, and this hypothesis is one that my team have been testing in the neuroligin 3 mouse model. So in order to do this, we turned to the touchscreen task battery and selected the rodent version of the continuous performance task, which was, at the time, the best translatable test for assessing attention in mice. This task involves training mice to respond to one rewarded stimuli and to inhibit their response to 4 unrewarded ones. This complex set of behaviours are not acquired by mice overnight. Akin to lengthy preparation required before running a marathon, our touchscreen mice are also carefully prepared. Mice are habituated to the reverse light cycle. They are nocturnal, and cognitive testing is best done when both mouse and human tests are in their awake cycle. Mice are food restricted to maintain motivation to learn. They are habituated to the touchscreen chamber and to the taste of milkshake. All of this before training begins. Once training is underway, they are monitored in real time. Their training occurs every day at the same time of day to avoid stress. Their food is titrated. Their weight is closely monitored. They're more work than the family dog. In early training, we place mice into touchscreens, and the first thing they do is explore the chamber. This innate exploration eventually leads to an interaction with the screen and the reinforced stimulus. This triggers the delivery of a reward, and for those wondering, the milkshake is the Australian brand Nippies, and we deliver 7 microlitres each time. It's the combination of this innate exploration and these low-effort conditions during training that form an association in these mice between response and delivery of reward. We iteratively introduce more complex demands on these mice, starting with simple visual discrimination, withholding their response to a simple distractor, and then the full set of 5 images, and subsequently we reduce the response event rate. Following training, we reduce the stimulus target duration to tax attention in a number of probe repeats, and we rebaseline sessions between. So we use signal detection theory to measure their performance, and in particular composite measures to assess their hit rate, false alarm rate, or their ability to withhold their response to the unrewarded images, D prime, or ability to discriminate signal over noise, and C, as an indicator of their liberal or their conservative response strategy. So early in training, mice were required to discriminate between 2 stimuli. This stage was conducted over 12 days, and most measures shown up here show evidence of task learning, including an increase in hit rate, a decrease in false alarm rate, improvements in D prime or ability to discriminate, while their strategy remains variable. Interestingly, NL3 mice show reduced false alarm rate early in training, and this was the first sign that we saw of a cautious response strategy. When we moved to the full task, mice were required to respond to 1 out of the 5 images. NL3 mice showed a reduced false alarm rate that resulted in an advantage in their ability to discriminate from signal to noise, and this was consistent across all our training days. We reduced the response event rate from 50% to 33%, and this is important because it is closer to the human response event rate. This pattern of responding in NL3 mice was maintained. When stimulus duration was reduced and attention accordingly taxed, NL3 mice lost their advantage, specifically at more difficult and shorter stimulus durations. They also increased their conservative responding. This was the first description of atypical attention in an autism mouse model. We have since reproduced this finding in another cohort of mice. While we can definitely draw some parallels between the human condition and these findings, we noted something in both studies that caused us to revisit our interpretation of the data. When mice respond correctly in touchscreens, we measure their latency to reward magazine, where the reward is delivered. NL3 mice were actually quicker to collect their reward in all stages of the continuous performance test. Could this be a sign of increased motivation for the reward? Reassuringly, we didn't find any differences in basic reward seeking, in body composition, or in multiple measures for metabolic chambers. However, none of these measures directly tell us about motivation. Could increased motivation actually be driving the increased performance in the CPT, or the continuous performance test? To be sure, we employed the progressive ratio test of motivation. This task is relatively simple in design, but not in effort, hence a measure of effort-based motivation. In fixed ratio training, the response outcome association is reworked in successive sessions to include graded effort. Mice move from touching the screen once for a reward to 3 to 5 touches. Once mice have formed this higher effort responses outcome association, they are progressed to the final stage, the progressive ratio task, where across the session there is diminishing reward return for responding. Mice are required to nose poke the stimulus on the screen +4 every time they show up. The point in which they stop responding is called the break point, and this is thought to represent motivation. And typically most mice will actually press the screen 50 times continually, over and over again, in a ramping difficulty before they give up. So what did we see in NL3 mice? We found slightly reduced responding in NL3 mice during early fixed ratio training, and a consistent decrease in break point in NL3 mice, indicating that they were less motivated to persist for a reward. Strangely, this was against our prediction, as we saw reduced reward latencies in NL3 mice during the CPT. So despite breaking earlier in the progressive ratio task, NL3 mice continued to stick their head in the reward tray to either similar amounts or even more than wild-type animals. They were still also faster to collect their reward. So we believe these mice were still interested in the reward, but perhaps they had a weaker association between the response site and the actual delivery of reward. We were perplexed and we felt we needed to replicate these findings before publishing, which is not always common practice in behavioural science. However, when we did this, we found no genotype difference. These two cohorts did share something very similar, which was the prior training in the continuous performance test. So to remind you, these mice were trained not to respond to 4 unrewarded stimuli, which is in stark contrast to being asked to nose poke a screen 40 times, 50, 60 times, and then do it all over again. And in the CPT, NL3 mice were better at withholding their responding during this task. And we believe this genotype-specific shaping of response inhibition is carried into the fixed ratio and the progressive ratio task, affecting the exploratory phase in the fixed ratio and responding during the uncertainty of the progressive ratio. And indeed, in a second cohort, we saw a similar phenotype to cohort 1. During fixed ratio, we saw reduced responding in NL3 mice. Another difference between these two cohorts was the length of fixed ratio training. In the second cohort, fixed ratio training was 3 sessions longer than the previous cohort. So differences in early training are commonplace in touchscreen testing, because we have certain criterion that specify that we move mice to the next stage altogether to reduce variability. So there are always a couple of stragglers, and in this case there were a few more stragglers, which delayed this part of testing or training. So extending the fixed ratio training led to the disappearance of this phenotype towards the end. And accordingly, when mice were progressed to progressive ratio following this slight extension in training, we saw no genotype effects in progressive ratio. Did this extended training help mice to overcome the carryover effects from the prior training in the attention task? At the same time, we had another cohort in training to follow up some interesting effects driven by methylphenidate, which I will not focus on today. This cohort was naive and not exposed to prior training, and spoiler alert, we saw exactly the opposite effect. So what's going on here? This naive cohort, when they were probed on progressive ratio, showed an increase in responding. And this progressive ratio increase was only from day 6 onwards. As you can see, they deviate towards the end. And so when we looked at the fixed ratio training, we saw that their difference emerged during later bits of the section. And so we suspected that NL3 mice might actually be sensitive to the effects of overtraining, because these emergent differences are indicative of habitual behaviour, patterns of behaviour. To test this in another naive cohort, we introduced 2 devaluation probes where mice were given free access to the reward 30 minutes prior to testing. Mice in habitual responding will maintain their response rate regardless of if they've had that reward devalued. In the first devaluation probe, NL3 mice responded more, indicative of higher habitual behaviour as we predicted, but by the second probe had reduced their responding levels to wild-type levels. This suggests that these devaluation probes led to a switch from more habitual to goal-directed behaviour in NL3 mice. And accordingly, no genotype difference was seen in the remainder of fixed ratio and also progressive ratio, supporting our hypothesis that this increased responding was driven by habitual responding. So with another 2 cohorts showing no differences to the mix, we were finally ready to move on. So I show you this tail because this is the hidden behaviour that you often don't see in the top tier publications. So I'd like to propose an embodied view of cognition to better explain the data I just showed you. So this view places a bigger emphasis on the body and motor behaviour in shaping cognition, and it argues that instead of a straight line from input computation to output, there are multiple driving factors that drive motor behaviours. And this could include prior learned behaviours, like in the continuous performance test, the carryover effect from this to cohorts 1 and 2, or habit formation that we saw in cohorts 3 and 4 in the previous study. These differential outputs will produce different feedback on the underlying computation. So we're not really measuring motivation in this case, but habitual behaviour. So noting there were no behavioural differences, we wanted to see if there were any gross neuroanatomical differences in the animals, and here is the only MRI data I will present today. My PhD student drew manual regions of interest throughout 30 brains, and I'd like to pause to appreciate his mammoth effort here. We saw volumetric reductions in key motor areas like the dorsal striatum and the substantia nigra. The dorsal striatum has been implicated in driving habitual behaviour. We didn't see any differences in motivational areas like the ventral striatum or nucleus accumbens, or indeed to task mapping areas like the prelimbic area or orbital frontal cortex. This supports our interpretation of habitual behaviour in these mice and warrants further investigation into the dorsal striatum, given its involvement with habitual learning. So to circle back to our findings from the continuous performance test and how they relate to autism, a few things stood out. We could now conclude that their discrimination advantage was driven by conservative, low impulsive responding, not differences in motivation. We had also undertaken a systematic review of the translatability of 3 commonly used attention tasks in mice and found that the continuous performance test did not fare well. Mice in this task do not behave like humans, seated in a chair, so we're unable to measure response times, which is a key measure in attention research. It's tricky to conclude if these mice are genuinely missing targets or if they're simply little chaotic animals. Most importantly, the continuous performance test measures sustained attention. And while this is a crucial component of the attention system, reports of differences in ability to orient attention, both in infants who are later diagnosed with autism, but also in autistic adults, point to this system being the most relevant. So the attention orienting system helps to direct our attention to stimuli and works with the alerting system to help us maintain our state of readiness, and also executive control system to determine the priority of salience. While these systems interact, their function is due to discrete brain regions and neurotransmitter systems. There is evidence of atypical attention orienting in autistic people, and here are 2 major pieces of work led by my PhD student, Xuting Li, including a systematic review and also a study in autistic individuals run by my collaborators and I. We do not know how the alerting system interacts with the orienting system in the context of autism, as this represents an ideal opportunity to repurpose attention modulating treatments and further understand how genetic variants produce changes. This knowledge gap warrants our attention. So attention orienting in humans can be assessed using the Posner task, and here it is here. So if you were a participant, you would be sitting in front of a screen and asked to fixate on this central cross. You would be asked to either click left or right, or saccade when you see the asterisk or target. In this simulation, you may have noted that the asterisk was first cued on the right and then invalidly cued when the target appeared on the left. Our response times are quicker when the cue predicts the location of the target, and this is the underlying principle of how the Posner task assesses attention orienting in humans. The difference in response time to valid and invalid trials is one of the main measures, and it indicates that the cues are being used. The cues, which you can see here in one of the boxes, can be changed in their probability and also in their look to measure two different types of attention orienting. A non-predictive flash is used in the exogenous or in the involuntary stimulus-driven attention orienting task. And colours indicating left or right, shown at higher than chance rates, so they can actually be predictive, are used for the endogenous or the voluntary goal-driven attention orienting task. So a major roadblock to my team assessing attention orienting in mice was the complete absence of a task. So following a major effort by Dr. Shu-Ting Lee in my team, we reverse translated the Posner task for use in mice. In the exogenous task, mice were trained to respond to a brief flash on the left or the right of the central fixation point. In this example, the mouse is first validly cued, and in the second trial, you'll note that it's invalidly cued, and you can see its whole body movement to correct its response, which correlates with an increased response time. For the endogenous task, a central left or right slanted grating was used to indicate the location of the task. And like the exogenous example, you can see that a slowed response is there for the invalid trial. The ability to perform this task has been replicated now in 3 independent cohorts. And my collaborator, Catherine Johnson, who works with children and has data from the endogenous orienting task, can attest to how difficult this version is. So we were quite surprised that mice were able to do it. So training a mouse to voluntarily fixate at the front of the touchscreen, or the equivalent of training a dog to stay, is no easy feat. We needed to do this so we knew that our mice were not running around, that they actually saw the cue, and that our response times were valid and easy to compare between animals. So we did this by slowly stretching out the wait time between fixation and target display over multiple sessions. And you can see here in this graph that there are no differences in ability to undertake this training in NL3 and wild-type mice. So we end up with 2 versions of the task. As you saw in the video, one with a peripheral cue for exogenous attention, and the endogenous task with an angled grating. During training, the target was presented at 1,000 milliseconds, or 1 second. In the probe session, we increased the difficulty of this task by including 500 milliseconds and 250 millisecond target durations. Like humans, all mice consistently showed better performance in valid compared to the invalid trials, which you can see here. This is our tick to know that our mice can perform this task. All mice quickened their response time and decreased accuracy as target duration reduced, suggesting that as the task becomes more difficult, the mice were rushing to respond at the cost of accuracy. And NL3 mice were most affected, showing much larger reductions in accuracy compared to wild types. NL3 mice had more difficulty responding to the short presentation of target, or difficulty in orienting their attention in more complex conditions. This finding now allows us to investigate if attention modulators can restore this to neurotypical levels. But what about the other task, endogenous goal-directed attention orienting? In this task, mice showed the expected quickened response times and decreases in accuracy with increased task difficulty, but NL3 mice performed similarly to wild types, indicating that goal-directed or endogenous attention is unaffected by the NL3 mutation. So the cholinergic system has been identified as the main system modulating attention orienting in humans, primates, and also in rats. So to investigate the involvement of this system in mice for the first time, we administered low and high dose mecalamelamine, which is a cholinergic antagonist. As we hypothesised, high dose treatment increased response times and reduced accuracy in the Posner task, but there were no differences seen between NL3 mice and wild-type mice. Confirmation of the involvement of the cholinergic system in attention orienting in mice really builds confidence that this task is actually able to capture a proxy that is relevant to the human condition. So our next steps are to utilise more specific pharmacological tools to further understand cholinergic involvement in attention orienting. I've shown you a discrete example of how we can use tools to investigate human-relevant cognition. What next? Advances in invasive neuroscience techniques allow measurement and control of discrete neural circuits in mice while they are performing behavioural tasks. Fibre photometry can capture population-level calcium activity from specific cell types within brain regions, and also can be integrated with optogenetics or DREADS to stimulate or inhibit specific circuits. In vivo electrophys can also be used to observe cell type specific activity but with higher temporal resolution. This work is complex and comes with many caveats, but many are showing that it's possible. So in order to know if you can use a mouse to resolve human brain structure and function, you need a valid mouse model, hopefully containing a high confidence gene candidate and reliably showing relevant cognitive phenotypes. This is not always simple, as diagnostic criteria are not always fit for purpose, and there is validity in modelling a unique facet, like what we have done with attention. Proxies are just that, proxies, but some are more comparable to human cognition. I've shared with you the process we went through to unpick if our mouse model was in fact more motivated by milkshake. This highlights the need for careful replication. I've also described the first task to assess attention orienting in mice, and the fact that we're able to demonstrate that a cholinergic antagonist modulated this suggests the same system is involved across species. This brings more confidence that we're able to study human attention orienting in a mouse. And then the last one I posed earlier, are mice really smart enough? I'm going to leave this one to Douglas Adams. I would like to thank my team, both current and alumni. Those directly involved in this research are currently listed here. I'm so proud of the effort, care and diligence my team bring to our mouse studies. This work is physically exhausting and also requires a great deal of creative thought in design. In particular, I would like to spotlight Ricky Dingwall, who led the journey to dissect the signal from noise in the conflicting motivation data. And he also traced all the brain slices. And he's a pretty master slide designer, so I've captured some of his slides in my talk. Thank you, Ricky. I'd also like to thank Xuting Li, who led the mPOSNA task development and subsequent studies with my wonderful collaborator, Catherine Johnson. Catherine is a psychologist, and part of my journey is really reflected from learning from her. I did not know what a cognitive construct was. I thought attention was what we needed to model. Thank you also to all my other collaborators who have contributed essential technical and knowledge expertise and great energy and enthusiasm. And a huge shout out to the Florey Ethics Committee, who run a tight ship and do everything in their power to make really good science happen. I must also mention the core animal services team. Without them, this work would not have happened, and of course, our mice. Last thank you to you. Thank you for being here and listening and attending today. It was a real honour.
Thank you, Emma, for the great talk. We have a few questions in the mobile app. I'm sorry for the AC too strong. Unfortunately, nothing we can do about that for now. So the first question is, there are quite some articles questioning the validity of mice behavior tests, particularly the Maurice water maze, which is a go-to in studies testing cognition. What's your take on that?
I hope that I address that by showing how fickle mouse behaviour can be. And I most certainly have done a lot of water maze testing myself, and I believe that cognitive testing using touchscreens absolutely trumps water maze. I'm not sure how we address that, because no matter what I do in my lab, it needs to be broader than that. So one group alone is not going to be big enough. So I wanted to have a shout out to the touchscreen community with 300 sites across the world, probably more now. And Tim and Lisa maintain their lab, and they have a real emphasis on open science, data sharing, and bringing a community together. So it's these sorts of grassroots movements, similar to what OHBM stands for, that I think will allow the field to shift.
Thank you. Another question. Actually, it's a very good one. So, is there a gold standard for defining mouse area homologous in humans? So, for example, how should studies of the frontal lobe be handled, which is developed differently in the two species?
I have watched full-blown fights around the ability to talk about prefrontal cortex homology between mouse and human, and it's really interesting. I hope we can keep debating this. Of course, there are areas that are involved in these cognitive tasks in humans that are just simply not in mice. And one I can point out is frontal eye fields, which are key to attention networks that are involved in attention orienting, but most certainly are not in mice. I think we need a combo of multiple different fields working together to be able to efficiently map. There are ways of doing this using the advanced neuroscience techniques as well that I mentioned, but it's lengthy work.
I think one of the interesting movements in mouse work is moving research towards more naturalistic settings or, less impoverished environments. I mean, in a lot of cases, what you do with a mouse is either the most exciting or terrifying thing that it's ever done, sometimes at the same time. And so how do you think this would compare, using the touchscreen to data one could capture in a kind of more naturalistic setting, whether it be a very large, enriched environment with RFIDs, when you think about social behaviors that you talked about before. Can you maybe comment on that?
Absolutely. I think the MPOSNA task in its design is more naturalistic because what it's doing is it's actually enabling a mouse to orient its attention based on what it would be doing in the wild. So it's freely moving and it's unfixed. Most attention tasks in mice require the mouse to be head fixed, and this is highly stressful and really unnaturalistic. So just from a concept of task design, I think we need to be thinking about real-world environments when we design our tasks. I started off my neuroscience career looking into environmental enrichment and have contributed quite a number of papers in this field. While I can see the benefits, it also introduces noise. And I think there's a really interesting field in mice emerging where you embrace heterogeneity, not just in genetics, but in multiple different labs, in different types of training, in different modalities. And I think that even though it was a lengthy and unusual story to tell in a keynote, the work on our motivation task shows that we were consistently able to get a behaviour coming through. If we had not tried to look at it from multiple different angles, we may have got the wrong conclusion. Real-world conditions are human conditions too, so as a community working with animal models, we have to keep talking about this.
So I'd like you all to join me in thanking Dr. Burrows for a fantastic keynote lecture, and enjoy the rest of your conference.